I'm Shannon Burnett, and my advisor is Jonathan Freeland, and my co-advisor for the MBM program is Martha Gillette. And today I'm going to talk to you about my project on characterization of the glymphatic peptidome by mass spectrometry. So I'll start with some background on the glymphatic system, because this is important for understanding my project. The glymphatic system is a waste clearance pathway in the brain and central nervous system. And this works by the fluid surrounding the brain actually flowing into the brain along the perivascular spaces surrounding the arteries. And the fluid is thought to flow through the brain parenchyma after it exits those perivascular spaces, and then it leaves the brain parenchyma along the perivascular spaces around the brain. So this could have many important functions, including clearance of waste molecules and fluid from the brain. And it could also be involved in neurodegenerative diseases, including Alzheimer's disease and Parkinson's disease, where the peptides amyloid beta and alpha-synuclein are known to accumulate in the brain. And this system is also, it's been found to be most active during sleep, which is pretty interesting and makes it important to consider circadian rhythm and time of day, which we have done in this project. So these are the main questions we're hoping to answer with this project. The first one is, which peptides are cleared from the brain by the glymphatic system? And the second one is, how do several factors affect this peptide clearance? And these are location, time of day, and circadian rhythm disruption. These are the methods we are using to hope to answer these questions. First, cerebrospinal fluid, or CSF, is collected by my collaborators in Martha Gillette's lab. And this is collected from, so far, just the cisterna magna of rats. But we are planning to also look at several other locations in the glymphatic pathway. And we're also looking at time of day. So how does the peptide profile of glymphatic fluid change over a 24-hour period? And we're also going to look at circadian rhythm disruption. So how might this affect the peptide clearance in the brain? After collection, I isolate the peptides using cell phase extraction, and then I perform LCMS analysis. LCMS is liquid chromatography mass spectrometry. This is an untargeted sensitive technique, which is important because we don't need to know which peptides we expect in the sample. And we're also able to detect peptides in these low volumes of samples. And how this technique works is through ionization, separation, and detection of the mass to charge ratios of these peptides. So this results in mass spectra, which is shown at the bottom. And through further data analysis, I'm able to identify these peptides. So we're using a peptidome approach, which means we're looking at the endogenous peptides in these samples, and this is done through de novo sequencing and database searching. And then we're able to compare the peptide profile of these different groups to find the effect of location, time of day, and circadian rhythm disruption on the peptide profile of this glymphatic fluid. So here's some of the results. First, we actually characterized the peptide profile of cerebrospinal fluid collected from the cisterna magna of rats without controlling for time of day. And this is to show that we can characterize these peptides. And these Venn diagrams here are showing a comparison of two different database searches, either UniProt or Peptide Atlas. And I'm showing the number of peptide spectrum matches, which are the spectra that matched a sequence in our database, and also the number of peptides identified in these samples for each database. So you can see that UniProt gave a lot more peptides, but it was also valuable to do the other database search as well, because not all of them are overlapping. I'm also able to characterize the post-translational modification profile of these samples, of the peptides in these samples. And you can see that phosphorylation, methylation, and acetylation are among the most common. We're also able to see what are the genes or proteins that these peptides are expected to be from. And you can see that there is some functional information from hemoglobin and fibrinogen, but there are also several interesting peptides which are expected to be involved in signaling at the synapse. We're also able to identify many de novo only peptides. These do not match the database, but they can be used to compare between the different groups and to find out if there's an effect of these different variables. So the next part of this is that we compared the effect of time of day. We looked at two different groups, midday and midnight. And this is all for cerebrospinal fluid that was collected from the cisterna magna of rats. And so glycemia time 4 and 15 are midday and midnight, respectively. And this shows the number of peptides identified for each of these two time points. And then of the 13 that were present at both times of day, I performed relative quantitation to see what the relative concentrations were. And 5 of them showed a significant difference. And you can see that these are proteins with different functions. So many different functions of these peptides were found. And this is gene ontology classification that I did to show this further. So of all the peptides that were identified, this shows there are many different functions that are expected. So here's a summary of these results. 133 peptides were identified in samples of CSF. 13 were in both groups, 5 with different relative concentrations. And there were 67 during midnight and 33 during midday. And a total of 105 peptides were differentially expressed between the two times of day. So some conclusion, there is a time of day effect on the peptide profile of cerebrospinal fluid. More peptides were actually expressed at night, which for rats would be consistent with the circadian rhythm that's been found for the glymphatic flow. So this flow it's been found to be at a higher rate during sleep, and this would be during the day for rats. So if there are more peptides, it's possible this is partly because the glymphatic peptide clearance is not happening at such a fast rate, but more work is needed to see if this is really the reason for this. And In the future, we are hoping to find the effect of more time points. So looking at a higher sample size and more time points for this time of day effect. And we're also going to look at the circadian rhythm disruption. So seeing how this may affect the peptide clearance in the brain. I'd like to thank several people. First of all, my lab, including my advisor, Jonathan Sweetler, Dr. Dharmesh Parmar, and the entire group. And I'd like to thank my collaborators, Martha Gillette's lab, and Wei Chen have collected most of these samples. So thank you. And I'd like to thank the MBM program, of course, and NIH NIDA for funding. Yes.
Yeah. Given it's such a very sensitive technique, you know, for such a large number of proteins to be completely missing, do you think they're missing? Do you think it's a sampling?
That's very true. We are working on increasing the number of peptides identified. We're also looking at the endogenous peptides. So that's not as many as you would typically find in a proteomics study where you're looking at the proteins, but we definitely would expect a higher number of peptides, and we're working on increasing the sensitivity of this method. Yes?
There's a question online. How many individuals did you take CSF from in each time group?
That's a great question. So there were only, I want to say 3 to 4 replicates in this group. This was, we're definitely going to do a lot more replicates in the future.